Now, let me explain proteinous part of the cell membrane. I explained that cell membrane is made of three chemical components. It's made of lipids, it's made of proteins, it's made of carbohydrates. Now, let me talk about proteins of the cell membrane. Generally, all proteins related to the cell membrane are divided into three basic types. The first type of protein is called integral proteins. Second type of protein is called peripheral protein. And third type of proteins are called lipid anchored proteins. Okay. Now, integral proteins are those types of proteins that cross all over the cell membrane from the inside to the outside. Okay. And these types of proteins consist around 25 to 30% of all proteins in the cell membrane. And when we detach integral proteins from the cell membrane, the cause is destruction of the cell membrane. Okay. And another type of protein, which is peripheral protein, these peripheral proteins are attached from the inside or from the outside to the cell membrane, and detachment of these proteins are easy for us. Okay, and they do not cause destruction of the cell membrane. Got it? And third type of proteins are called lipid anchored protein. These are types of proteins that are connected to the lipid or to the one leaflet of the cell membrane from the inside or from the outside of the cell. For example, they are called GPI anchored protein that I'll explain all of them one by one. Now let's talk about integral proteins. Now, integral proteins are those types of proteins that cross over all the cell membrane. Basically, we have two types of integral proteins in our cell membrane. Some types of integral proteins are called single pass integral protein. They pass through the cell membrane one time. Okay. And another type of integral proteins are called multi pass integral proteins. Multi pass integral proteins. Okay. And these span through the cell membrane more times or more. Okay. Now, a, a transmembrane protein, because integral proteins are also called transmembrane proteins. A transmembrane protein has three parts. Its one part is hydrophobic. It means it locates inside the cell membrane. Okay, and the two parts of uh, transmembrane proteins are called extra membrane parts, or they act as a receptor and effector domains. For example, this is a multipass, or this is a single pass transmembrane protein. From the outside, this inside of the cell, this is outside of the cell. From the outside, this protein or this domain of protein will bind to a ligand or to a stimulant. Okay, and this part is called receptor. Okay, and acts as a receptor, and it receives signals. Okay, this is transmembrane part or hydrophobic part of this protein. This inside part of this protein causes uh, causes production of different effects inside of the cell. That's why this part is called effector domain. For example, this ACE1 pass receptor or serpentine receptor that we'll talk about it in future lectures. Okay, this effector domain of 7 pass receptor binds in the inside of the cell to a peripheral protein which is called G protein. Okay, so it causes production of different effects inside of a cell. Now let me explain different types of integral proteins and their related different functions. For example, we have a type of integral protein which acts as a channel. Okay, and as a collectively they are called channel proteins. So they are channel proteins. So we have different channel proteins, and their role are to help in transmission or to help in transportation of different substances that they are not lipid soluble across the membrane, and we will study about it in cell membrane transportation. For example, this is a sodium ion. Okay, we have more concentration gradient of sodium outside of the cell than the inside of the cell. Now this sodium can flow through these channels. Okay, and we have different types of channels for this sodium or for potassium or for calcium. For example, here is a type of channel which is called leaky channels. Okay, leaky channels are those types of channels that they are always and always leaky and permeable for ions. Okay, and ions move through this channel from high concentration gradient to low concentration gradient. For example, if this channel be for sodium, so sodium will move from high concentration gradient to the low concentration gradient. If this channel is for potassium, so potassium moves from the inside of the cell to the outside of the cell because we have more concentration gradient of potassium inside of the cell. Okay, so this is called leaky channel. We have another type of channels in the cell membrane, and they are called mechanosensitive channels. It means these channels are sensitive to pressure, okay, to mechanical pressure. For example, we have ends of neurons in our skin, and these are called noise receptor, and these are located in our skin and for the purpose of pain receiving receptors. Okay, now when you press or touch or put pressure on these receptors, now this gate is semi-closed. Okay, but when you put pressure on this receptor, this gate will be opened, and now this channel is permeable for specific ion. For example, if this channel is for sodium, so now sodium flows from high concentration gradient to the low concentration gradient. We have another type of channel proteins, and they are called ligand gated channels. Ligand gated ion channels. Okay, it means a substance will come and it will bind to a specific portion or to a specific region of this channel, and this channel will be opened by this ligand. Okay, now due to this ligand, this channel will be opened, and now this channel is permeable to a specific ion, and a specific ion will move from high concentration gradient to the low concentration gradient. And we have another specific type of channel, okay, which are called voltage gated ion channels. That we will talk about this type of channel in upcoming video lecture under the title of depolarization and repolarization. But anyway, for example, we have some cells, okay, that in these cells we have specific voltage inside of the cell. For example, in a muscle cell or in a neuron, we have a specific voltage that are from negative 70 up to negative 90 millivolts. Okay, now if you change this intracellular voltage, if you add positive charges to the intracellular area and you bring the intracellular membrane from negative 90 to negative 70, or from negative 70 to negative 50 or 55, so this changing in voltage causes opening of this channel, and that's why these channels are called voltage gated ion channels. For example, we have a sodium voltage gated ion channels in many cells and calcium voltage gated channels in many cells. Got it? So these were all about channel proteins. The second type of protein in our cell is called receptor proteins. Okay, these receptor proteins act as a receptor. For example, this is a 7-pass receptor. 7-pass receptor because it spans through the cell membrane 7 times. Okay, or it's also called serpentine receptor. It means it's snake like, or it's also called G protein coupled receptor because they join to a globular protein which is called G protein or globular protein. That's why this is called G protein coupled receptor. But anyway, a substance, a neurotransmitter, a ligand will come and it will bind to a specific domain of this protein, which is called receptor part. Okay, and now this protein will be stimulated and it causes production of a lot of events inside the cell. That's why they are called receptor proteins. Okay, because they act as a receptor. So this was the second type of protein in our cell membrane. The third type of proteins in our cell membrane are called carrier proteins. They carry something from the inside of the cell to the outside of the cell, or from the outside of the cell to the inside of the cell by transformational change in their structure. Okay, they will change their shape and they will transport something from the inside to the outside of the cell, or from the outside of the cell to the inside of the cell. For example, this is a protein, a carrier protein, which is called GLUT or glucose transporter. Now, for example, in this step 1, it's in this shape that its outer area is available for glucose molecule. Now, it will catch glucose, okay, and it will bring glucose inside it, and then it will transport glucose by changing in its shape. Okay, that's why they are called carrier proteins. Okay, and we have many types of carrier proteins in our body that we will study about all of them in upcoming video lectures. The fourth transmembrane or integral proteins are called enzymes. Okay, because they act as an enzyme in our cell membrane. For example, they convert a substance A into substance B. Okay, that's why they are called enzymes. And we have an example of this type of protein in our cell membrane, and it's called adenylyl cyclase. And you will know about adenylyl cyclase when you study about 7-pass receptors. Got it? And fifth type of transmembrane and integral protein is called linker proteins. They are transmembrane proteins and they link the intracellular proteins to the extracellular proteins. Okay, and they act as a backbone of a cell membrane and they give strength to the cell membrane. Okay, this is the connector, the cytoskeleton of the intracellular area to the extracellular proteins. And we have different types of this type of proteins. For example, a protein which is called dystrophin in a skeletal muscle. So this was all about integral proteins. And we have many types of proteins in our body for different purposes, for different functions. Okay, now about peripheral protein. Peripheral proteins are attached from the inside or from the outside of the cell to the cell membrane. And a great example of peripheral protein is G protein, which is connected to the G protein coupled receptor or 7-pass receptor and acts as a signaling protein. And also some types of proteins that are connected from the inside of the cell to the cell membrane to give strength to the cell membrane. For example, two types of proteins which are called spectrin and ankyrin in RBC or in red blood cell. Okay. And lipid anchor proteins are some types of proteins that they have some specific functions. But anyway, integral proteins are the most important proteins in our cell membrane. Got it? Now let me talk about carbohydrates. Okay. Now let me talk about carbohydrates of the cell membrane. Carbohydrates surround our different cells for different purposes. Okay. And they're a thin layer around our different cells. Okay. So let me explain some important functions of carbohydrates. Its one important function is that carbohydrates act as a glycocalyx, or they are going to make glycocalyx. What do I mean by glycocalyx? Glycocalyx is a narrow layer around our cell membrane, okay? And this glycocalyx protects our cell membrane from different types of damages, okay? Another important function of carbohydrates is that they have a role in ABO blood group system that we've studied them in RBC, okay? But anyway, for example, this is an RBC. In some types of RBC, we have some types of carbohydrates. For example, they are called N-acetyl N-acetyl galactosamine, okay? And some other types of herbicida, for example, only galactose, and the causes to make ABO blood group system. For example, our blood group system depends on these carbohydrates, that it's A blood group, or this is B blood group, or this is O blood group, and we will study about them in upcoming video lectures. Don't worry. Another important function of carbohydrate is that it has rule in adhesion. It means this is one cell, this is cell number A, this is cell B. Glycocalyx or carbohydrate comes out from this cell and also from vessels, and they will be connected to each other and causes adhesion and connection of two cells. And also a lot of proteins and other materials help these two cells to be connected to each other, okay? Another important function of glycocalyx or carbohydrates are that they have role in signaling, okay? They produces different signals to our immune system, and say for our and tell for our immune system that this cell depends to our own body, okay? So that's why it's also very important, okay? And also carbohydrates have role in immunity, okay? For example, when a normal cell is going to a cancer cell, okay? So this cancer or carcinogenic cell will produce some different types of carbohydrates and gives signals to our immune system that this cell is destroyed or this cell is not normal, okay? So these were different functions of carbohydrates. But when we add carbohydrates to the lipids, okay, so this is called glycolipid. But when we add carbohydrate to protein, this is called glycoprotein. And we have different types of glycolipids and glycoproteins around our cells for these different purposes and functions. Got it? So these were all about carbohydrates. Now, let me explain and tell you some related terms to fluid mosaic model, okay, or some new terms to the last design of our cell membrane. First of all, our cell membrane is made of lipid. Secondly, it's made of proteins. Thirdly, it's made of carbohydrates. and we studied different types of carbohydrates, proteins, and lipids. Okay, now here is some terms related to this fluid mosaic model that I explained it previously. For example, here is a concept of asymmetry versus sidedness. Sidedness means that we have two different and distinct parts of cell membrane. For example, this is a cell membrane. The inner leaflet of the cell membrane is different structurally and functionally from the outer leaflet of the cell membrane. This is called sidedness. It means we have two different structural and functional leaflets of the cell membrane. For example, an inner leaflet, we have phosphatidyl serine and phosphatidyl ionocyte, and in outer leaflet we have phosphatidylcholine and sphingomyelin. Okay, so they are different in structure and in functions. And also in inside we have different peripheral proteins, and in outside of the cell we have different types of proteins. And asymmetry means that structurally and functionally inside of the cell is different from the outside of the cell. Sidedness means two different parts of a cell membrane, and asymmetry means two different parts of a cell membrane functionally. Okay, it's structurally, it is functionally, but anyway. Play-flow mechanism means that some lipids have this capability to dive to the inner leaflet or from the inner leaflet to the outer leaflet. Okay, it's not very important. We have a new idea in fluid mosaic model. It means that fluid mosaic model has fluidity. Fluidity. What do I mean by fluidity? Fluidity means that cell membrane has laterally movement, or these lipids in our cell membrane have laterally movements. Okay, for example, when a thing goes out from the inside of the cell to the out, or from the outside of the cell to the inside of the cell, which is called endocytosis or exocytosis. So during exocytosis, for example, this is a cell. Okay, this is a cell. Now I want to export something from the inside of the cell to the outside of the cell. Now I can transport this substance through a process which is called exocytosis, that we will talk about it in upcoming video lecture. Now this exocytosis causes production of holes in our cell membrane. Okay, so that's why when we have fluidity, okay, lateral fluidity in our cell membrane, by the help of this fluidity we will pull this holes once again, or these holes once again. So that's why this fluidity is very important. Now here is some factors that influence our fluidity in our cell membrane. For example, temperature. A high temperature causes fluidity to be increased. Okay, saturation of fatty acids. Unsaturated fatty acids or unsaturated phospholipids decrease fluidity in our cell membrane. Okay, for example, saturated fatty acids are straight. Okay, they're straight, but unsaturated fatty acids have bends in their structure. So when we make more bends, okay, when we make more bends, so it means we made a lot of spaces between these feet of phospholipids. So that's why it causes the fluidity to be increased. Got it? And third factor which influence our fluidity, it is cholesterol. It is cholesterol, because we have cholesterol molecules between these fatty acids. So when we increase cholesterol molecules, okay, they will fill these gaps. Okay, and finally fluidity or lateral fluidity decreases. Okay, so that's why these factors are a little bit important, because with increase or decrease of fluidity, cell membrane will lose their normal functions. Got it? So now let me explain some important functions of the cell membrane. Its first function is that a cell membrane has rule and protection of a cell. For example, this is a cell. This cell membrane surrounds our intracellular structures, our DNA, our nucleus, and anything which are present in our cell or inside of our cell. Okay, so all of these materials are protected by the cell membrane. Its another function is it has role in structural support of a cell. It means it gives strength to our cell, okay, and supports our cell structurally, okay. Its another function is it has role in cell membrane transportation. Cell membrane transportation, okay, in cell membrane transportation, because it's semi-permeable. It means it allows something to go from the outside of the cell to the inside of the cell, or from the inside of the cell to the outside of the cell, okay. Its another function is that it has role in cell adhesion. For example, this is one cell or cell number A, this is cell number B, so these two cells will be connected through the cell membrane or by the cell membrane, okay. Its another function is that it has fluidity, which I explained it previously, and also it has flexibility. It means when we put some pressure or when we press a cell, it prevents from rupturing of a cell, okay. And also it has role in cell signaling through different types of receptor proteins, through different types of carbohydrates, and so on. And the thickness of the cell membrane is around 5 to 8 nanometer, okay. So we studied a lot about cell membrane structure and its functions, okay. For example, we studied about different components of the cell membrane, we studied about different cell membrane models, we studied about different types of lipids, proteins, and carbohydrates, and so on. So please don't forget to share, to like, and to subscribe our channel and our videos. Have a good time till next lecture.